Welcome everyone to our webinar series. It's now 3 o'clock Eastern, so we wanted to get started. This is the fourth in our series. My name is Marie Latulippe, and I work with some of the nutrition programs of ILSI North America. And this series is brought to you in collaboration with the American Society for Nutrition. And what we're doing is we're exhibiting the ongoing work of faculty and students at the University of Illinois. And all that work focuses on the gut-brain axis and the gut microbiome. So just another usual reminder that everybody's on mute. Please type your questions in the chat box at any time. And we are recording the webinar, so we'll post that on our website afterwards as the data allow. Just a minute on ILSI North America. For those that aren't familiar, we're a nonprofit scientific organization, and we advance food safety and nutrition science for the benefit of the health of the public. We like to remind everyone that we do not lobby. All the intellectual contributions are from scientists that are in industry, government, and academia. We combine that with pooled funding to allow us to advance science. So we just wanted to thank everyone for joining us today and for all the webinars in our series. And our audience today is reflective of that tripartite approach that we undertake in all of our activities. So if you'd like to learn more about the organization, feel free to email us, visit our website, connect with us through Twitter or LinkedIn. And with that, I am pleased to introduce our first speaker, and that is going to be Dr. Aditi Das. And Dr. Das is a membrane protein biochemist. She completed her Ph.D. in chemistry at Princeton University. And currently, Dr. Das is an associate professor in the Department of Comparative Biosciences and Veterinary Medicine at the University of Illinois. So Dr. Das, we will turn the platform over to you.
Yes, first of all, I would like to thank ILSI and Nutritional Sciences, especially Dr. Elvira, for inviting us and giving us this opportunity. And I've been really enjoying the webinar series myself and listening to the other talks, and we hope that all of you enjoy it today as well. So today I'm going to talk about some new lipid metabolites. Those are formed from the omega-3 and the omega-6 fatty acids. These lipid metabolites are neuromodulatory in their function. And we have further gone and collaborated with Dr. Stillman, who's going to give us the talk about the different animal models in multiple sclerosis and how that helps us to study about the role of these novel omega-3 metabolites further. And then Justin will tell you about the real study that he has been doing. So I will get started. I would first talk about how omega-3, omega-6 fatty acids are converted into these novel lipid metabolites in the body and how they have very interesting pharmacology. So all of you have heard all the time, and you listen to, you're all nutrition researcher, you know that omega-3, omega-6 fatty acids have been implicated in very beneficial effects, harmful effects, but they are known to play very important roles in cardiovascular diseases, cancer, neurodegenerative diseases, etc. So as a biochemist and as a lipid biochemist, my interest and my laboratory's interest is essentially to find out why. Like, why is the omega-6, omega-3 fatty acids causing all these beneficial effects, and what is their biochemical route through which these effects are seen? So what happens when you consume omega-6, omega-3 fatty acids from your diet is that these are esterified and stored safely into your membranes. You can change the lipid composition of your membrane depending on if you're on a completely omega-3 diet or an omega-6 diet, or even on a saturated diet. When there's any kind of inflammation or any kind of upregulation, such as maybe a disease, then what happens is the phospholipase is upregulated. So this is an enzyme that releases these free fatty acids. For instance, the arachidonic acid is omega-6 fatty acid, and icosapentaenoic acid and DHA, docosahexaenoic acid, are the omega-6 acids, not omega-3 fatty acids. So what happens to them further is they are metabolized by enzymes in our body to form prostaglandins, leukotrienes, and epoxides. While you may or may not be familiar with the cyclooxygenase and the lipoxygenase metabolites, that's prostaglandins and leukotrienes, I'm sure all of you at some point have taken NSAIDs. NSAIDs block the cyclooxygenase pathway because that blocks all the pro-inflammatory metabolites. But the body itself is very clever. It makes its own anti-inflammatory molecules from these omega-3 and omega-6 fatty acids, which are called the epoxides. And all of these are anti-inflammatory nature. Who makes these epoxides? These are made by cytochrome P450 epoxygenases. This chart shows here that the human body has 57 cytochrome P450s, and all of these P450s can have the potential to metabolize lipids such as the PUFAs into their corresponding epoxides. Especially the P450s of the CYP2 class, which are found in the endothelial cells, are known to metabolize these PUFAs into their corresponding epoxides. So essentially what they do is these P450s are involved in lipid metabolism, and they convert any omega-3 or omega-6 fatty acids, such as arachidonic acid here, into these epoxides. And all these epoxides, both from the omega-6 and omega-3, are anti-inflammatory in nature. They are vasodilatory. They are good for the cardiac health, so they have lots of good properties. Now I'll shift gears to another very important class of compounds, and we are familiar with this plant. It's becoming more and more important, and this is a cannabis plant. For 5,000 years, we have used cannabis, and this is first report of cannabis from 2700 BC in China, where cannabis was essentially used for treating everything. And then in the Indian Vedas, cannabis is mentioned, and there is also a cannabis plant called Cannabis indica variety, which is popularly used in India and has a lot of CBD content. So the first scientific literature of the cannabis use came from Dr. J.R. Reynolds in this paper on the therapeutical uses and toxic effect of cannabis indica in Lancet. He was the physician of Queen Victoria who suffered from migraine and a lot of pain, and cannabis, she really needed cannabis to take care of them. So in the recent times, several publications came. People are still curious to know, how does cannabis really work? What does it do? There have been several review papers and research papers showing cannabinoids can control spasticity and tremor in MS model. And that's why one of our interests, why we work with Professor Stillman to see the effects of some of these lipid molecules in multiple sclerosis. And then there's also a lot of study that point out to the anti-inflammatory nature of the cannabis molecule. Let's delve into a little bit into why cannabis molecules or phytocannabinoids that we derive from the plant are anti-inflammatory. So medical marijuana, or let's say cannabis, has this component called THC, and there are other 80 to 90 minor cannabinoids which people don't understand what they do. But one of the things that have been shown that they do is bind to cannabinoid receptors. Cannabinoid receptors are found throughout the body, and they have been known to involve in many processes in the body. And some of these are reducing pain, reducing tremor and depression, spasticity. They have many, many different effects. And they're also involved in retrograde signaling. So two approved cannabis-based drugs is Sativex and Sesamet, and it has been shown that cannabinoids have neuroprotective property as well as immunomodulatory property. But then scientists started asking, why do we have cannabinoid receptors in the body? They're highly present in the brain. And then they started looking, there must be endogenous lipids or endogenous molecules in the body that bind to and activate these cannabinoid receptors. So that led to the discovery of the endocannabinoids. So they are endogenous cannabinoids in 1992 and 94. That's anandamide from the Sanskrit word ananda, which means happiness. So the more of these molecules you have, the happier you are. And 2-arachidonyl glycerol. And both of these molecules are strong agonists for cannabinoid receptor 1 and sometimes 2. And they also have similar properties like the phytocannabinoids from cannabis, which is they are also neuroprotective and has been shown to have immunomodulatory properties. But one of the things about the endocannabinoids is that they are lipid-like, and so are the cannabis molecule. They are lipid-like as well. Where do we get these endocannabinoids in our body from? So now we are back to the food component. So the omega-6 endocannabinoids, if you look at their structures, are derived from the omega-6 fatty acids, arachidonic acid. But in 2010, people got curious, we are consuming all these DHA, brain is supposed to have a high percentage of DHA. Do we really have omega-3 endocannabinoids? And that started the research field of starting to look for omega-3 endocannabinoids. And our lab got very interested in these omega-3 endocannabinoids and their further metabolism. So the goal was to bring the best of two worlds, that's the world of cannabis and omega-3 together. And now we have new molecules, which are cannabinoids as well as omega-3 fatty acids. And these are omega-3 fatty acid endocannabinoid. But we just learned that the omega-3 fatty acids, they get converted by the enzymes to form these epoxides. So how about we bring the best of three worlds? Like we have cannabis, we have omega-3 fatty acid and epoxide, and all three of them come together to have cannabinoids, omega-3 fatty acid, and epoxides to form these complicated molecules, which are now omega-3 fatty acid endocannabinoids, and they're also epoxides. So today my talk is actually going to focus the tale of the two lipid metabolites that we discovered, and these are synthesized by cytochrome P450. So cytochrome P450 is the author of this book. So let's first look at the first omega-3 endocannabinoid epoxide. The epoxygenases, which are the cytochrome P450, were first hypothesized to convert this DHEA, which is the omega-3 ethanolamide, into the different regioisomers or the epoxides. The next thing we wanted to show is, do we really find this in the body? So for that, we had to develop a mass spec method. This is MRM, and this is a targeted lipidomics method to look for specifically these particular metabolites. So once we had this specific method ready, then we extracted these metabolites from the rat brain, and to our surprise, we found that the levels of some of the epoxides are same as anandamide, which is the most well-studied endocannabinoid that was discovered in 1992. So when we looked at the other organs, we not only found it in the rat brain, we also find it in pig brain, mice brain. We also found it in heart. Kidney has a lot of these molecules, spleen and liver, and especially the organs that are more rich in cytochrome P450, epoxygenase, and kidney being one of them. So then we wanted to see if these are enzymatically formed. They are formed by the purified cytochrome P450 as well as brain microsomes. And usually they form the terminal epoxide ethanolamide preferentially over the other epoxides. We then wanted to see the pharmacology of the molecule because we wanted to know, yes, these are found in the body, they are synthesized, but what do they really do? So for that, we also worked in collaboration with other groups. So we wanted to first look at its interaction with cannabinoid receptor. So as I mentioned before, there are two types of primary cannabinoid receptor, one in the central nervous system that is supposed to be responsible, like the interaction or agonism of this receptor is responsible for the psychotropic effects of cannabis molecules, as well as the neuroprotective effects and anti-pain effects. On the other hand, the cannabinoid receptor 2 is mostly expressed in the immune cells, and they have anti-inflammatory actions. And CB1 is the most highly expressed GPCR in the brain. So then we wanted to study that what is the interaction of these omega-3 endocannabinoid epoxide with this receptor. And we found another very interesting result. So while the omega-3 endocannabinoid by itself was not a strong agonist of either CB1 or CB2, you can see these are pretty high, but the epoxidation of these molecules made very potent cannabinoid receptor 2 and sometimes cannabinoid receptor 1 selective agonist. So what might be happening, and this is at least, of course, at the hypothesis stage, is that these molecules are produced due to an inflammatory trigger, and then they crosstalk with different cell types which have cannabinoid receptor 1 and cannabinoid receptor 2. So then we started collaborating with Professor Rodney Johnson's group, who were expert in microglia. And microglia is the resident immune cells in the central nervous system, and they mediate neuroinflammatory processes. They play a very key role in chronic and acute neuroinflammation diseases such as MS, Alzheimer, Parkinson, Huntington, etc., many neurodegenerative diseases. It has been shown, because microglia expresses CB2 ligand as opposed to CB1, that we can have a CB2 selective agonist which can have a therapeutic potential for both controlling both pain as well as immune system modulation. And because we saw that our molecules, once they are metabolized, can form CB2 selective metabolites, therefore they can interact potentially with the microglial cells. So using an immortalized cell line called BV2 microglial cells, which can show two types of phenotype. Of course, there are many intermediate phenotypes, just not a pro and an anti, but it goes from pro to anti. But you look for few genes in the pro-inflammatory state and few genes in the anti-inflammatory state. And we were able to show that these molecules were anti-inflammatory in nature just with respect to the fact that they increase interleukin 10 and arginase 1. So these were first indication of these molecules, but we needed to study this further. In collaboration with the Stillman group, we isolated the primary microglia from mice. So this is the litter bond. This is 2-day-old pup, and you dissect the brain and you get the primary microglia mixed culture. You can isolate the primary glia from that. And we were able to show that what we saw in the immortalized cell line with BV2 repeats, and there is an increase in the interleukin 6 with these omega-3 endocannabinoid epoxides. So now we are at the step that where we want to know more about these new molecules in a more physiological system and in vivo. So we are collaborating with Bruce Hammock's lab to look at the anti-pain behavior of these molecules. So today, Justin Kim, he will talk about the behavior of these molecules in a multiple sclerosis animal model. And we have previously looked at these omega-3 endocannabinoids in osteosarcoma, and we were able to show that these molecules slow down cell proliferation. And these molecules were all vasoactive, so they are antiplatelet aggregatory as well as they do vasodilation. So they have many favorable pharmacological properties. So now I will quickly go to the next anti-inflammatory molecule that we worked in the laboratory, which is omega-6 endovanilloid epoxide. I'm introducing another term here. What is endovanilloid receptor? So this is transient receptor potential cation channel, also known as TRPV1. So TRPV1 is a very interesting channel because this is one of the important channel in pain regulation. TRPV1 is found in the gut, and the gut is lined with TRPV1, and it's not known what is all the TRPV1 doing in the gut. The other place that you find the TRPV1 is in nociceptive neurons, DRG neurons, or you can say sensory neurons, and they are involved in the transmission of pain, also known as nociception. The other interesting part is these DRG neurons or sensory neurons also have a lot of lipids. So TRPV1 is activated by all these different kinds of molecules, and agonism of TRPV1 causes pain, such as capsaicin, and we have all had it, very high heat, and PGE2 or some sort of an inflammatory agent can activate TRPV1. So therefore, we are looking for how does the body manage its own pain? Like, does it have its own molecules or lipid molecules that can be antagonists of these TRPV1? So some of the endocannabinoids discovered previously by Vincenzo Di Marzo's group and other groups showed that anandamide is a TRPV1 antagonist, while NADA is a TRPV1 agonist. So NADA is a dopamine lipid derivative. So it's a specialized molecule. It has a dopamine group in this, you can see it in the blue here, and a lipid. And then on the other hand, we have N-arachidonyl serotonin, which is a serotonin group and has the lipid tail. Originally, this N-arachidonyl serotonin was discovered and isolated from intestines. They are found in the gut. And there is a researcher, his name is Renger Wittkamp, whose lab really looks at what are these molecules doing in the gut. He has also isolated the DHA versions of the serotonin molecule, and that's also found in the gut. And gut is also lined up with the TRPV1 channel. So there is a mystery there. And there is also the question, like, who generates this molecule? And are these the gut microbiome that generates these molecules and these lipid molecules? But I will talk to you today about the metabolism of these N-arachidonyl serotonin with P450 and how they become very strong antagonists of TRPV1. So what we are going to explore is the TRPV1 cannabinoid receptor 1 and 2 axis. This axis is involved in pain directly or indirectly. So for instance, CB1 activation, or let's say CB1 agonism, is known to be anti-pain. CB2 agonism is thought to be anti-inflammatory, so that in turn might control pain. And then TRPV1 antagonism is definitely anti-pain. So we are looking for a favorable pharmacology of a molecule that is an agonist of CB1, agonist of CB2, and an antagonist of TRPV1. So what we do is these are the molecules that we are studying. And as I said, it's bringing the best of three worlds together, which is that lipid moiety, which is the lipid chain. And then you bring in the dopamine and the serotonin molecule, as well as the epoxides. And this, firstly, we were able to show that this does happen. The cells have a capacity to convert them into the epoxides. We then looked at its antagonistic behavior, and I'm going to focus only on N-arachidonyl serotonin epoxides. So this we found was a very strong antagonist of TRPV1 channels, and this could be seen in their this is the IC50 value, where it shows that this epoxide molecule is more potent, almost 70-fold stronger antagonist of TRPV1 compared to the non-epoxide version of this molecule. So in order to further study this molecule, we collaborated with Professor Hongzhen Hu at Washington St. Louis, who is an expert on pain and itch, and he helped us discern the pharmacology of these molecules further. We did calcium imaging of dorsal root ganglion neurons, which he isolated, and DRG neurons expressed TRPV1. So what we were able to show is this 1415 EPO-NASH, as we call this, was really able to inhibit the capsaicin-induced response in DRG neuron cultures. And this shows here the data in real time. And then using electrophysiology, we were able to show, like, while the vehicle shows action potential firing, the N-arachidonyl 5-HT or serotonin shows lesser action potential firing. But when you have the EPO NASH, which is the epoxide version of N-arachidonyl serotonin, you don't see action potential firing at all, and there is a depolarization. So this can be seen clearly on the data on the right, that this is a strong antagonist of 14-15, strong antagonist of the TRPV1. What was the other good properties of these molecules was this was anti-inflammatory, and it was also a CB1 full agonist. So this molecule itself has a very interesting pharmacology, which makes it a potential scaffold to develop an anti-pain drug. So now I'll come to the conclusion of what I wanted to talk about, and give way to Professor Stillman and Justin, who have very interesting view on how to study these molecules further, how this can really help to understand not only their pharmacology but help in developing drugs, and even to understand what do omega-3, omega-6 fatty acid really do in the body. So the conclusion for the first part, which is the omega-3 endocannabinoid epoxide, is that these generate from the crosstalk of the endocannabinoid and the CYP pathways, and they have very differential ability, and they can really activate wide range of receptors, including both CB1 and CB2, and they were shown to have anti-neuroinflammatory property, which we wanted to study further in an in vivo model. They were vasodilatory, anti-angiogenic, and also exhibited anti-cancer properties. Then we looked at the omega-6 endovanilloids. Then these are lipid neurotransmitter conjugates. We know that cannabinoid receptor and TRPV1 axis is involved in pain. These endovanilloid epoxides are formed from NADA and NASH, and both of the NADA and the NASH are found in the brain, in the gut, many parts of the body, and they are known to be TRPV1 agonist and antagonist. So we found that one of the epoxide metabolites of the N-arachidonyl serotonin was a strong antagonist of TRPV1, a full agonist of CB1, and also anti-neuroinflammatory. And we were able to confirm some of the results in a DRG neuron cell culture using electrophysiology and calcium influx study. So our conclusion from this study was these endovanilloid epoxides can act as potential scaffold to develop anti-pain drugs, which are partial or modulators of different receptors. So one of the concepts in this field is, so far whenever we develop drug, we develop strong antagonist or strong agonist of receptors or enzymes. But the time has come when we do not want to shut down or switch on completely, but we want to have a rheostat control on the receptors, because TRPV1 is needed, but we still need to reduce pain. So now I will thank the people who helped me with all the work. One of the things I've realized being a faculty is all about who you work with and who help you out. So these are my current collaborators, and the work I presented today was in collaboration with Professor Rodney Johnson, Stillman, and Professor Fan. And I did not talk about any of the biophysics work that my laboratory does today. And I want to most of all thank the people who do the work. So these are my students, and this is COVID time, so we are all on Zoom. I want to thank the four people whose work I presented. That is Daniel McDougall, who's did the omega-3 endocannabinoid epoxide work, William Arnold and Lauren Carnivale. They did the work on the endovanilloids, and Josephine Watson on the CB1, CB2, and Justin Kim will show his work on, translating this work into an animal model. And I thank the funders very much for believing in the science and funding it. And finally, I want to thank all of you. We are what we eat, so we have to be careful about what lipids are we eating, because we are generating an array of lipid metabolites in our body, which can have a varied pharmacology. So thank you very much for listening to me, and I'll take any questions.
Great, thank you so much, Dr. Das. I think in the interest of time, we'll go to Dr. Steelman. And so I would like to introduce Dr. Steelman while he gets his slides up. Dr. Steelman is an associate professor in the Department of Animal Sciences, and a large component of his current research, it focuses on understanding how systemic inflammation resulting from upper respiratory viral infections exacerbate symptoms of multiple sclerosis. So he's also investigating the therapeutic potential of nutritional supplementation to alleviate neuroinflammation. So with that, we would like to turn the platform over to you, Dr. Steelman.
Thank you very much. I hope everybody can see my slides.
Yep, we can see them.
Wonderful. So I'm glad that I've been given this opportunity to speak about multiple sclerosis and some of its animal models. I was very excited to learn about the research that Professor Das has been doing. Her molecules seem to be have a lot of therapeutic potential for this disease that I'm going to tell you a little bit about. And so I hope that you guys will appreciate this. So thank you very much for the introduction. As you may know or may not know, multiple sclerosis is a neuroinflammatory and a demyelinating disease of the central nervous system, confined to the central nervous system. It's thought to be autoimmune mediated with both T cell and B cell involvement. T cells being the pro-inflammatory phenotypes, Th1 producing interferon gamma, and Th17 cells producing interleukin 17, and also GM-CSF. Approximately 2.5 million people in the world are affected by multiple sclerosis. The onset of the disease is pretty cruel. It usually happens in the second or third decade of life. The disease is chronic and there's no cure for the disease. Rather, as we'll find out, the disease progressively worsens. The disease has a female to male predilection, so females are almost twice, if not 4 times more likely to contract MS than males. The cause is unknown, as I said before, but potentially there's a viral component or trigger coupled with genetic predisposition that would unfortunately allow somebody to develop MS. So from a pathological standpoint, the disease was first described by Jean Cruier and Robert Carswell in the mid-1800s. These were both pathologists working on understanding atrophy and various causes of disease. But while they described the lesions that occurred in MS patients, they didn't see the patients themselves, so they could only predict what the lesions might have caused. Carswell is most famous with his watercolor drawings of MS pathology, which are shown here. You can clearly see that the lesions can occur in the cerebellum and also in the spinal cord and are pretty widespread and confined to the white matter. Jean-Martin Charcot is credited with defining MS as a disease and discovering MS as a disease. And the reason is because he was able to link pathology with clinical symptoms. He identified disease in living patients, and he was able to describe lesions that occurred around vessels and the expansion of these lesions from the ventricles. And this is consistent with the idea that multiple sclerosis is an autoimmune disease caused by the immune system coming from the periphery into the central nervous system parenchyma. In fact, if we were to look microscopically at a lesion, which I've stained here with a marker for myelin red and a marker of inflammation or microglia green, we can clearly see a defined lesion edge, and within the lesion itself, it is packed full of microglia or gliosis, and along the lesion edge, there is increased microglial reactivity. If we were to stain this lesion for T cells, we would find that there would be T cells within the lesion as well as along the lesion edge, with CD4 cells staining most prominently within the lesion and CD8 T cells staining along the lesion edge. If we were to stain the tissue for axons, neuronal axons, we would find that there's quite a lot of axonal preservation within the lesion itself, and that the lesion is not devoid of axons, even though it might be reduced in axonal number. So that brings up the idea that we can still promote repair or remyelination within the lesion and perhaps regain recovery of function. The disease is very characteristic in terms of its natural history. Even though between patients it can be heterogeneous, the natural history is pretty conserved. And it's so much conserved that Douglas Firth was able to diagnose Augustus D'Este posthumously after he had died. And the reason was because Augustus, who was the son of King George III, kept a very meticulous diary. And in his diary, he wrote about having experienced optic neuritis, where he lost eyesight in one of his eyes. And then over time, that went into remission. And then a few years later, it reappeared. And so we see that there's a relapsing, remitting disease episode that transitions into a progressive deterioration of symptoms or of function. That's very characteristic of MS. Looking at it now, we know that there's quite a lot of subclinical inflammation that occurs within the brain, which is shown at the bottom. And when this inflammation reaches some critical threshold, the patient may experience a relapse or onset of disease, which could be called a clinically isolated syndrome. So the patient may wake up one day and not be able to move an arm, and that might trigger them to go to the neurologist. Or they may wake up one day and not be able to see. These symptoms, which would constitute dissemination between time, different periods of life, and space, which would be different areas of the brain, would make the neurologist consider multiple sclerosis very much as a diagnosis. So 85% of the patients will exhibit a relapsing remitting disease course for quite some time. And then over time, the number of relapses will fade, and instead the patients will start to undergo a period of secondary progression, which is characterized by continual worsening without relapses. And interestingly enough, there's not as much inflammation that's occurring in the brain at this time frame, but there's quite a lot of axonal loss and decrease in brain volume. So these are phenomenon that would definitely lead to permanent loss of function, where there's no repair that could be facilitated. So how does this damage happen? What is pretty much well established is that microglia within the brain parenchyma will become activated. And once these microglia are activated, they'll start to produce cytokines and chemokines that act on the brain endothelium to upregulate adhesion molecules that will attract autoreactive T cells to diapides across the endothelial barrier. But they get stuck in this perivascular space by the glial limitins, which is held together by these astrocytic infi. They'll accumulate to form a perivascular cuff, and for some reason they can overcome this barrier and come in contact with a resident antigen presenting cell, which may be a dendritic cell or a macrophage or even a B cell. Where they're presented their antigen, they become reactivated, and in conjunction with microglia and other antigen presenting cells, they'll start to produce toxic factors that will cause oligodendrocyte cell death and eventually neuronal cell death. And this will result in permanent loss of function. So B cells are also involved in this process. We know this because some of the most effective therapeutics for MS currently inhibit B cell function. So what's the cause of MS? We know that there's a genetic component, because if we were to look at people that have MS and, for instance, monozygotic twins, if one of them has MS and the other one is at least 30% likely to get MS over the course of their lifetime, but it's not 100%. And as we decrease genetic similarity, the risk for developing MS within a family is decreased quite substantially. So there's definitely a genetic component. And if we were to ask what is the genetic component that's associated with MS, the strongest allele is, strongest association is with MHC class two alleles, which are the way that macrophages present antigens to CD4 positive helper T cells. So that sort of makes sense since this is considered an autoimmune disease. However, it's likely that MS is polygenic and that not only the presence of HLA-DRB2-1501 would confer complete risk. And even in patients where 100% of genetic identity is shared, so monozygotic twins, we see that the risk rate is only 30%. This sort of indicates that genetics can't play an entire role here, and that there must be an environmental influence. So for the longest time, there's been the idea that there's an infectious cause of multiple sclerosis. And the reason that we think this might be true is we can certainly see inflammatory demyelination brought on by experimental infections with various viruses, including Tyler's murine encephalomyelitis virus, murine hepatitis virus, same Linky-Forest virus, canine distemper virus, measles virus, and there's also some reports that SARS-CoV-2 can cause encephalitis and acute disseminated encephalomyelitis. So we know that viruses can cause inflammatory demyelination. There are also epidemiological studies, such as those that occurred on the Faroe Islands by John Kurtzke, that indicate that perhaps there's an infectious cause to multiple sclerosis. And this is quite interesting. So the Faroe Islands are located between Europe and England. And during the World War II, England stationed troops on the islands at these points in order to keep the Nazis from invading. Now, what's also important about this is that prior to 1950s, there was no recorded cases of MS on the islands. And then in 1950s, John Kurtzke noticed that there were outbreaks of MS that occurred at these locations. So he was able to superimpose where the outbreaks of MS occurred with where the British troops were stationed, and found that there seemed to be quite a strong correlation between where the troops were stationed and where MS had an outbreak, or where people developed MS. So the idea is that perhaps the British were able to bring some sort of infectious pathogen to the islands and expose the native people who were living on the islands to this pathogen, and then they were able to develop MS, unfortunately. There's also been isolation of many viruses from the central nervous system of MS patients, but we haven't been able to pinpoint one as the infectious cause of MS. The strongest association currently is with human herpes virus 4, or Epstein-Barr virus. All MS patients have had Epstein-Barr virus. The incidence is about 100% versus 95% of the population. And there's some other suggestion that this virus can trigger MS onset. Viruses are not only thought to play a role in the onset of MS, but they're also been shown to play a role in disease exacerbation. And this was first shown by Sibley and Foley in about 1965, where they noted that MS patients who had symptoms of upper respiratory infection were more prone to relapse. And these data have been revisited over the last several decades and are pretty reproducible, indicating that infection by upper respiratory viruses may trigger relapses, although no one is quite sure how this occurs. You could envision that since the immune system can be modulated by infection, it could trigger autoreactive T cells to diabetes across the barriers and trigger disease exacerbation. So there's been some influence of the microbiome as well, and definitely some interest since 2010. So here I've plotted the number of publications that have arisen after the search term microbiome and MS, and you can see that there's quite an explosion of papers. So I was very happy to find that Mirza just published a recent systematic review on studies concerning the microbiome and MS that were published between 2008 and 2018. In total, they identified 415 articles, and from these, they were able to exclude all but 10 by 2 independent reviewers. There were a total from these 10 articles of 286 MS cases. Most all of the MS cases had relapsing remitting MS, which we would expect, compared to 296 controls. And what they found screening these studies is that diversity was not different between groups, neither alpha. In fact, alpha diversity wasn't significantly different in 8 of the studies that examined alpha diversity. Beta diversity was significantly different in only 2 of the 7 that included beta diversity measures. But they did find differences at the taxa level, and one of the ones that's most striking is the Akkermansia mucinophilia. This bacteria seems to promote Th1 cell responses, and the studies that have shown that this bacteria is involved in MS indicate that perhaps it's promoting inflammatory Th1 responses, which could contribute to disease exacerbation. So how is it that we study MS? Because the etiological agent is not established, there's no perfect model for this disease. There's no animal model for human MS that completely recapitulates the disease process. So we're at a disadvantage here. However, several pathological features can be reproduced using animal models. These include autoimmune-mediated animal models, such as experimental autoimmune encephalomyelitis, remyelination models. So in these models, mice are given toxicants such as cuprizone or injected with toxicants such as like the lysolecithin, and these will cause a focal area of demyelination that can be followed over time to look at therapies that might improve repair. And finally, there are viral models of inflammatory demyelination, some of which I mentioned earlier, such as Tyler's murine encephalomyelitis virus. These viruses are very beneficial in understanding how viruses can trigger autoimmune inflammation, and so very important in determining the pathogenesis. By far the most commonly used animal model is experimental autoimmune encephalomyelitis. In this animal model, mice such as C57 black 6 mice or SJL mice are injected with encephalidogenic peptides such as MOG 35 to 55 for C57 black 6 mice, or PLP 131 to 151. These are myelin peptides, and they're emulsified in a very strong adjuvant. So once the mice are immunized with these myelin proteins, they will develop an inflammatory demyelinating disease that's characterized by inflammation in the spinal cord and can be scored relatively simply by watching their gait. So a score of a 3 might indicate rear limb paralysis. A score of a 1 would be tail paralysis, and they can be followed over time. So here I've been showing the relapsing remitting model of MS, where SGL mice are immunized with PLP, and they will develop an onset characterized by remission and then relapses. If we were to look in the spinal cord of these mice, what we would find is that there are huge lesions that are both demyelinating, are full of CD4 positive T cells, have astrogliosis, and also are full of microgliosis, similar to the lesions that are found in MS patient brain. So EAE itself is a CD4 T cell mediated disease. The encephalidogenic T cells, or the T cells that can cause disease, most potently produce IL-17, interferon gamma, and or GM-CSF. The disease can be actively induced by active immunization with peptides, or by transferring autoreactive cells to naive mice. So the transfer of autoreactive CD4 cells is sufficient for having disease onset. The pathology of EAE, as I've mentioned, is characterized by demyelination, reactive gliosis, and T cell infiltration. So it is a fairly decent model with which to study neuroinflammatory processes of autoreactivity. So with that, I'd like to thank my group before we hand it over to Justin, and he can tell you about putting together the molecules and the disease model.
So starting with one related to, Justin, what you just spoke about. So the question is, can you achieve the benefits that you observed with dietary omega-3 fatty acids, say 3 to 4 times a week, or could you speculate on that?
Yes. So I think we can see very similar effects, and I think that overall it'd be very beneficial. I think something that we didn't really discuss a lot today is the fact that these downstream metabolites are produced very locally, and they can only be produced locally in high abundance if we have an abundance of the omega-3 fatty acids already in our system. So I think overall it's really important to look at to make sure that we're consuming more omega-3 fatty acids as a whole, because then we'll be able to see many of these same effects that we are discussing here today.
Okay, and then kind of a follow-up to that is it possible that you see the results, at least in part, from the production of resolvins?
Yeah, that's a great question. So typically we think about omega-6 fatty acids and omega-3 fatty acids as good or bad. Omega-6 fatty acids being bad and omega-3 fatty acids being good. In reality, however, there are a lot of other things that's going on with these molecules. So the omega-6 fatty acids are really important for the production, as we discussed, of the omega-6 variety of the endocannabinoids. But there are also prostaglandins that, for example, that can be produced by the omega-6 fatty acids. And this is really important for the first immune response. So not all inflammation is really bad. It's just the chronic inflammation that is bad. When it comes to the omega-3 fatty acids, and I guess specifically in terms of the resolvins, a lot of these effects could be attributed to the resolvins because they would be produced also by these essential fatty acids. But again, I think this kind of relates to the balance of omega-3 and omega-6 fatty acids that we're consuming in the diet to make sure that, again, it's in balance and we are not over-consuming one over the other.
So I wanted to add to this question. So in our studies, we don't look at resolvins, but we did look at resolvins in the study on the pig brain with Rodney Johnson, and we were unable to find a correlation with the omega-3 fatty acid, cognitive development, and resolvins. And also the levels, there is no resolvins ethanolamide, and our study focuses on the DHA ethanolamide. And I don't think there's anybody who's reported a resolvin ethanolamide. So yes, they might be forming, but from the DHEA directly, the resolvins are not forming here.
Okay, thank you. And going back to the dietary question, I guess, so Justin, you provided the endocannabinoids through injection. Is it just more direct than a dietary administration? Would there be any difference there that you could speak about?
Yeah, that's a great question. So I think the reason why we are doing intraperitoneal injections as opposed to a dietary supplementation is first to really ensure that we're administering the proper amounts and to make sure that it's circulating in the body. And then the second component, and I think arguably just as important, or if not more important, is that we, after doing the DHEA studies, we're also planning on looking at the DHEA epoxide studies, which are a little bit more downstream. Unfortunately, however, the epoxide is susceptible to being broken up into hydroxys or dihydroxy. So in place of the epoxide, there are actually two hydroxyls that will be at that olefin position, and these are very inflammatory actually. And this degradation occurs under acidic conditions, such as the stomach, which we don't really see anywhere else. I think in terms of future work, ultimately that is sort of where I envision this project going, where we are looking at how we can create a formulation that will make sure that the molecules are being properly digested, absorbed, and into the system, but is not being degraded by the acid in the stomach. But I think for now, our focus right now really is just looking at the roles of these molecules, both DHEA as well as the epoxide, and to prevent that degradation, we are conducting intraperitoneal injections.
Great, thank you. This question kind of goes back to Dr. Das, your opening talk, just describing endocannabinoids. So the question is, do levels of endocannabinoids in humans have a range? Does this relate to why we say some people have a different pain tolerance than others?
Yes, that's true. And there are several studies that shows that. So humans have different anandamide levels, and most of the studies are done with the omega-6 endocannabinoids. So I'm going to recall those studies. So yes, people have different pain tolerance. Like there was a woman who never felt any pain, and she actually didn't have the enzyme that degrades fatty acid amide hydrolase. And later it was discovered. So she never felt pain for almost 60 years, and they realized that her levels of anandamide are skyrocketing in her body. So there is a correlation with pain and these levels, yes.
Great, thank you. So this is a question for Justin. Can you talk a little bit more about the potential role of the omega-3 endocannabinoids as it pertains to the glial crosstalk?
Yeah, within the central nervous system, there exists a glial crosstalk between the microglia, for example, the microglia, the astrocytes, and the oligodendrocytes. A lot of the studies that we have looked at so far is focusing on the microglia aspect. So there are two things that kind of come to question here, considering that these are cells in the central nervous system, is one, are these molecules even getting into the brain to elicit their effect on the glia? And there are studies that do show this. The most notable one, I think, is a study where they had radio-labeled molecules such as the ethanolamide moieties of the essential fatty acids, and they actually show that it traffics into the brain through an intraperitoneal injection. And I think the second part here is the fact that it could also be synthesized locally. So we know that there's a high abundance of omega-3 fatty acids, and it's essential for brain growth and development. So how is it being produced locally? Is it produced in the endothelium? Could it be produced by certain glia? And currently, another area that I am pursuing right now is looking at how astrocytes may be producing these molecules. So data I haven't shown today does suggest that there is an increase of the cytochrome P450 epoxygenase enzymes in the astrocytes. So our next step is looking at the actual production of these molecules that's being produced and thus secreted, the DHEA epoxides that is. And if that is also happening, then that would influence the microglia. And overall, this interaction would then also influence the oligodendrocytes, which are very important for the myelination and remyelination of the myelin in the central nervous system. So collectively, this will also contribute to the myelination aspect of multiple sclerosis.
Great, thank you. So going back a little bit to the oral versus injection administration, the question is, how long do these endocannabinoids last in circulation, maybe for both of those routes?
Yeah, so in terms of sort of endocannabinoid circulation, so as I mentioned, they are typically locally produced. However, they can also travel. These molecules can be degraded pretty rapidly under the presence of hydrolases. So for example, the epoxides can be degraded by soluble epoxide hydrolase, and the ethanolamide can even be degraded by fatty acid amide hydrolase. In terms of, from the best of my knowledge, in terms of the degradation, the time frame, I am actually not quite sure. However, I do know that they are degraded pretty rapidly, and as a result, that also contributes to the difficulty in being able to detect these molecules and actually being able to study these molecules due to its, one, the low amounts of the local production, and then two, the quick degradation. With that said, however, I think it's really important to also mention that these are very, very potent molecules. So if they're really potent, then they don't really need to stick around for a very long time, and they don't really need to be produced in, like microgram quantities locally in the tissues. So hopefully that answered that question.
Great, thank you, Justin. Okay, we're getting to the end of our time today. I just want to make sure anybody has any other comments to add before we wrap up. Okay, so we just wanted to thank the three of you, Dr. Stas, Dr. Steelman, and Justin, for sharing your work with us today. We encourage everybody to visit our website to register for the final webinar in this series, which is next Friday. And during that webinar, we'll hear about this specific study, Strong Kids 2. And this is focusing on childhood nutrition and obesity and the microbiome brain axis. So with that, we'll wrap up for today. Thank you again for participating, everyone, and we hope to see you next Friday.
Thank you very much. I appreciate everyone's attention.
Yeah, thank you very much. And this was a great opportunity to co-present with my student, and that's a pleasure. Thank you very much.
Great, thanks. Have a good afternoon.
Bye.